All right, good morning everyone. Today we'll be discussing the renin-angiotensin-aldosterone system of the kidneys. So the key players in the renin-angiotensin system that we'll call RAS are the juxtaglomerular cells. And these cells function to sense low blood pressure. They're a special type of smooth muscle cell, and they are found near the glomerulus in between the afferent and efferent arterioles. According to Kurtz, their location is a juxtaglomerular position at the entrance of afferent arterioles into their glomeruli. So as we can see here in the figure, the juxtaglomerular cells are called granular juxtaglomerular cells, and they're located right at this junction in between the afferent and efferent arteriole. Juxtaglomerular cells, or as we'll call them, JG cells, their job is to release renin. And they are triggered to release renin by three different stimuli. The first is the cells can sense low blood pressure in the vessels. And once they sense the low blood pressure, they will release renin. The second method by which they release renin is through sympathetic nerve stimulation. As we can see here in the figure, the sympathetic nerve endings end on the juxtaglomerular cells, and when they are triggered, and these nerves fire after a major stressor, they trigger the JG cells to release renin as well. And the third means by which the JG cells release renin are through the macula densa, and they are cells that are found within the distal convoluted tubule of the nephron. And these cells sense low sodium concentration, and that's due to low blood pressure. So there's low fluid movement through the nephron, and therefore sodium is being absorbed. By the time the filtrate gets to the macula densa cells, there's a low concentration of sodium, letting the cells know that there is a low blood pressure and it needs to be raised. So the macula densa will then release a local messenger called prostaglandins that then act on the JG cells, and the JG cells release renin. Well, what is renin's job? So renin and angiotensin are two hormones that work hand in hand. Renin's job is to raise the blood pressure, and also to activate the essential hormones needed in this process. So liver cells make angiotensinogen, and angiotensinogen is a hormone precursor, and that means it's an inactive hormone. It's not targeting anything, it's not acting anything, nothing is happening with this hormone, it's just available in circulation. Renin comes along and converts it by cutting a portion of the angiotensinogen and turning it into the active hormone angiotensin 1. Angiotensin 1 is just one of the intermediates, and we need to convert it to angiotensin 2. And this conversion takes place by blood endothelial cells and with the help of the enzyme ACE, or angiotensin converting enzyme. And this angiotensin converting enzyme is made in the vascular endothelium of the lungs. Okay. So the ACE enzyme cleaves the angiotensin 1 and converting it into angiotensin 2. With patients who have issues with high blood pressure, there are ACE inhibitors that we will see later on in our discussion that stop the ACE from working on angiotensin and converting it to angiotensin 2, which stops blood pressure from being raised. So what are the effects of angiotensin 2? Well, angiotensin 2 works on 4 different cell types. the first of which are the smooth muscle cells in blood vessels. So when angiotensin targets these cells, they contract or constrict, and it causes increased resistance in the blood vessel, raising the blood pressure. Angiotensin II also acts on the kidney cells and causes them to retain water. And as they retain water, volume increases and we have a high stroke volume. Angiotensin II also acts on two glands, the pituitary gland and the adrenal gland. When it affects the pituitary gland, it causes it to secrete the hormone ADH. Now, ADH, it also targets some of the same cells that angiotensin II targets. It targets the smooth muscle cells in the blood vessels and causes them to constrict. Same as angiotensin II, increased resistance, increased blood pressure. ADH also acts on the kidney cells and causes them to retain water as well, increasing the stroke volume. Angiotensin II also acts on the adrenal gland, causing it to secrete aldosterone. And aldosterone is a hormone that acts on the kidneys and makes those cells again retain water, increasing the volume and stroke volume. So angiotensin either is causing a direct change, increase in blood pressure, or causing the release of hormones that eventually cause a raise in blood pressure. So as we'll see now, we'll get a brief overview of this RAS angiotensin system. How does angiotensin II work, and how does the angiotensin converting enzyme work, and how does that play into the medical necessity of needing patients to have a lower blood pressure.
Angiotensin II is an oligopeptide that circulates in the blood of many animal species. It is involved in constricting the blood vessels, an event known as vasoconstriction, thereby increasing blood pressure. It is an integral element of the renin-angiotensin system, which includes both the kidneys and the liver. Angiotensin II is produced from two precursors, angiotensinogen, produced primarily by the liver, and angiotensin I. The enzyme renin, released by the kidneys, cleaves the peptide bond between the leucine and valine residues on angiotensinogen, creating the 10-amino acid peptide angiotensin I. Subsequently, angiotensin conversion enzyme, or known as ACE, found primarily in capillaries in the lungs, cleaves the peptide bond between histidine and phenylalanine, resulting in the 8-amino acid long peptide angiotensin II. The active site of ACE, or angiotensin conversion enzyme, includes a positively charged zinc ion shown above. Each contribute to attracting the substrate, angiotensin I in this case, chemically to the active bonding pocket. While anchored in place, water can be introduced, resulting in the cleavage of the peptide bond as shown. A class of synthetically created organic molecules prescribed to reduce high blood pressure are known as ACE inhibitors. They function by preventing angiotensin conversion enzyme from creating the vasoconstrictor angiotensin II from angiotensin I. They perform this action by simulating the same bonding patterns exhibited naturally, a bonding pattern described previously. As a result, they often possess shapes and contain chemical charges analogous to those possessed by angiotensin I. A competition therefore takes place between ACE inhibitors introduced into the body and naturally occurring angiotensin I due to the fact that both are attracted to the active site of ACE. By occupying the active site of ACE before angiotensin I, ACE inhibitors effectively diminish the production of angiotensin II. This in turn can help a patient suffering from excessively high blood pressures maintain control over the renin-angiotensin system.
Okay, so that's an overview of the renin-angiotensin-aldosterone system. As we said, the key players are the juxtaglomerular cells that release renin,